Hi, this short lecture will introduce you to the Pet Organism Characterization project that we'll be completing over the next few weeks of the course. And I'll reiterate some of the things I already sent you in the email over the weekend. So each student receives their own pet bacterial species to characterize. I've already assigned you yours. You'll make observations and perform tests during the next several weeks. And as I explained, I'll be making those tests for you, taking photographs, and then sending you the results. The result data should be recorded in your course pack. I believe that's on page 72. So you can record the results there, as well as recording them in each lab exercise as we go along. And then in another week or so, there will be a spreadsheet that will be available to you where you can record that information as well. Because we will use all of that information from the class, from everybody enrolled in Biology 207, for the unknown characterization project that will be taking place the last part of the semester. You should learn to say the name of your pet. Jack, you have Staphylococcus aureus, or otherwise known as Staph aureus. Olivia, you have Klebsiella pneumoniae. Ben, yours is Escherichia coli, or commonly known as E. coli. And Yijin, you have Pseudomonas aeruginosa. So get to know the names of your pets. You should learn the risk group and biohazards, if there are any, associated with your pet. I did put on Moodle that bacterial pet chart, and it includes the risk groups, so you can look that up. The risk group is particularly important if you're actually physically working with the bacteria. You would want to know what risk group your bacteria is in and what are the signs and symptoms of infection, of working with that bacteria. And you can look this up just by Google, just a short description of signs and symptoms of infection. And you can record that on page 32 of your course pack. One of the first things to do with pet organism characterization is to look at the broth culture characteristics. A clear broth tube like this, if I can get a good photo here, well, that's not showing up very well. An uninoculated tube of media will just be clear. You can tell that nothing's growing in it. But once we introduce bacteria to that broth tube, it becomes cloudy or turbid. But there can also be other characteristics associated with that broth depending on the organism. Some may make that broth kind of slimy. Some of them will have bits floating throughout and are described as flocculent. Some form a ring at the top, known as a pellicle, and some will sediment. The bacteria settles out to the bottom. Next week, I will send you photographs of your bacteria growing in broth. The other thing you need to do is know the gram stain characteristics of your organism. I posted those photos on Moodle this weekend, so you can look that up for your organism. So what is the gram reaction? Is your pet gram positive or gram negative? What is the shape and the arrangement of your bacteria? And then estimated size. That will be difficult for you to do, so you can ignore the estimated size. But the gram reaction information would be recorded on page 33 in your course pack. I'll be maintaining your pet organisms. One thing we'll do is have a plate grow at room temperature and another at 37 degrees. Sometimes bacteria grow differently at different temperatures. So next week I will be sending you photos of the different of your plates grown at those two temperatures. And you can begin to tell if there's any differences, if growth temperature actually matters for your bacteria. So you should be able to start by doing that gram stain analysis and also look up the risk group and pathogenicity that's associated with your pet.